Today we will discuss about this effect of TGF-beta on glycosylation. So we will first go through the types of glycosylation that occurs in the mammalian cells. Though we have covered that thing many times in our previous videos, it's a good idea to revise those things before directly go into the effect of glycosylation on TGF-beta or the TGF-beta effect on the glycosylation. So there are mainly two different kind of glycosylation: N-glycosylation and O-glycosylation, and that will lead to the formation of N-glycans and O-glycans. N-glycans are usually branched on the asparagine residue of the proteins, and the sequence of asparagine residue should be NXT. That is, X can be any amino acid except proline. So here we can see that the branched glycans, N-glycans, that are expressed on this asparagine can further be divided into three different categories. And first one is this oligomannose, in which case the high mannose are expressed on the core structure. The core structure is common, which can be found in all the three categories, and this core structure is 2N-acetylglucosamine and 3 mannose. And in the case of this hybrid, there is a hybrid formations of the mannose and the this galactose along with this N-acetylgalactosamine, and the complex formation is contain this galactose and an N-acetylgalactosamine. Along with that, it will also contain this N-acetylnuramic acid or sialic acid, and it might also has this fucose. So these are the three different categories of N-glycans. And in terms of the O-glycans, here the commonly expressed O-glycans are shown along with those that are found in the cancers. So these TN, STNs are usually found in the cancers, in which case this N-acetylgalactosamine along with the fucose is called STN, and without fucose is called TN. And these core 1 structures are where N-acetylgalactosamine is attached to galactose, and in the case of these core 2 structures, the galactose is attached to another galactose, and on the other branch, it is beta 6 branch, it is attached to N-acetylglucosamine. And for the core 3 structures, only this N-acetylglucosamine is attached through beta 1,3 angles or beta 1,3 bond. And core 4 structures where this glucosamine is attached to galactose on both the angles, that is beta 1,3 angle and beta 1,6 angle. So these are the different kind of N-glycans and O-glycans that are formed. However, apart from that, there are other glycans are also found that are found on the lipids or on the cell surface membrane. We can say that those are these glucosyl ceramide and galactosyl ceramide. Along with this, the GAGs are also found. These are usually formed on the cellular matrix. So this is a brief overview of how these different kind of glycans that are found in the mammalian cells. So now, how this TGF-beta affect those glycan formations. So the actual final outcome is not shown here. The triggering point are shown here. How TGF-beta gets activated. The first way is that through this latent associated proteins that are found on the cell surface, the TGF-beta interact with this latent associated protein to form this latent associated protein or latent TGF-beta binding proteins. And in this case, the TGF-beta is in the inactive form, and then it can this proteases, it can undergo this inactive or latent TGF-beta become active. With the help of this protease, it will degrade the latent associated protein, and the TGF-beta will become free. And the other way is through the interactions with this integrin. So this traction with integrins will make the TGF-beta available or free. And once the TGF-beta is free, it can interact with this TGF-beta receptor 2 and receptor 1 and then activate those TGF-beta receptors through intracellular phosphorylations, and that will cause this triggering of the SMAD pathway. That is SMAD 2-3 phosphorylations will occur, and then it can interact with this SMAD 4, and this complex can translocate in the nucleus, where it can interact with the cofactor or transcription factor, thereby results in the epigenetic changes, and those epigenetic changes will cause this epithelial-to-mesenchymal transitions. This is how this TGF-beta affect the glycosylation. So once this transition occurs from epithelial to mesenchymal, and these things are also being supported by change in the glycosylation. So those change in the glycosylations need to be studied in detail to find out how this glycosylation plays a role in this epithelial to mesenchymal transitions. We will discuss the further informations that they have provided in this review paper in our subsequent videos. That's all for this video. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.